The next speaker is Professor Dr. Euverink. He's from, well, he was from Wetses, and I read he's from the University of the Institute for Technological Things in Groningen. He did some research on biofouling and the measurement of biofouling, and that's a very interesting study because at this moment we know that biofouling, of the biofouling potential affects the regrowth in distribution areas of drinking water supply. But if we change something and we want to measure if it's different, then we have this standard tool that takes half a year to get a result. And if it's not that good, well, another half year. But now Mr. Euverink is giving a new tool which makes it thus very easy, I hope. The floor is yours.
Thank you very much for your nice introduction. Actually, the institute where I'm working for is the Institute for Technology and Management at the University of Groningen. I'd like to present some results on a project which we did in the framework of SAWA. And SAWA is a sensoring network which was established in the north of the Netherlands. It's a collaboration of a lot of different small sensor companies. And we are trying to address all kinds of sensoring issues in the distribution network, so after the purification of the water. Also, the intake water is of importance, so we have a project on surface water monitoring, the quality of the water in the distribution network, and all kinds of different parameters like temperature and pressure and flow, particle analysis in these in the tubes. And also, very important, as Harry just mentioned, is the regrowth potential of the water. It doesn't make sense if you purify your water and you put it into the distribution network, and then you will have a lot of bacterial growth, and then the water comes at the customers with a lot of turbidity. That's not something you want. We actually have a demo site in Ingham. That's the building next to it. It was a pilot plant building from the Water Laboratorium North. It has been rebuilt into a sensor test center, and we have the different water qualities available for the different companies to measure the real water which is available. So my take-home message is that the analysis of the regrowth potential of water can be improved using a combination of nanotechnology and image analysis. And in the end, you can build an online sensor with this. Well, Harry just mentioned my sheet. It takes a long time before, with the current methods, to measure the biofouling potential or the regrowth potential. The two current methods I'm aware of is the assimilable organic carbon inside the water, and that's actually a technique which takes 10 to 12 days before you have some results. It's not always correlated with the real regrowth potential, so it's a parameter, and it gives you a clue that there is something wrong. And the other method, it's what they said at Water Laboratorium North to me, takes about 30 days to 3 months before you have any results. And that is a method where you put glass rings inside the distribution network, and you have a lot of glass rings inside the water, and once in a while you take out a glass ring and you analyze the biofilm on the glass. And then you need several time points before you have an accurate measurement. You need fast results if you want to optimize your treatment scheme. So then I thought, well, how can we do this? How can we actually address bacterial growth in a fast way? Well, I came up with a method like this, is that we have a kind of a flow cell where the water is flowing underneath a flow cell, and then the nutrients inside the water will diffuse through a small chamber on top of the flow cell. So outside the water, you inoculate bacteria. Doesn't matter which one. You can have indicator bacteria, you can have recombinant DNA bacteria, you can have special color bacteria, any strain you want. Because it doesn't come into contact with the water. So it's outside the system. So the nutrients which are necessary for growth of the bacteria will diffuse through that layer, and then the bacteria will start to grow. And the only thing you have to do is to analyze the bacteria on the chip. There's no flow cell between the microscope and the bacteria. So you don't have any disturbances with fouling of the flow cell. You just have to put a microscope outside your water and analyze your bacteria. Well, we analyzed some of the different barriers we could use, and we came up with the porous aluminium oxide. This is a very nice material. It is very porous, so nutrients can diffuse freely from the water to the bacteria. It's inert, so the bacteria have no problems growing on this material. It's flat. It doesn't have a fluorescence or an imaging background by using the microscope, so you can have nice and clear pictures. And because it's flat, you'll always have a nice sharpness of your picture. And you can do this automatically and not by hand. It's low cost. You can do some microengineering with it. It's biocompatible, so different types of bacteria grow as fast as on standard agar media. And for microbiologists, you can also grow organisms which are not able to grow on agar. So for instance, if you use this kind of material, you can grow a lot more organisms than on standard agar plates. It's low cost, and the only drawback in this system is this kind of a brittle material. If you take one of this if you take a material with a tweezer, you have to be very careful because else it will break. Well, this material actually exists. This is a platform technology from a company called Microdish. This is a startup company from the University of Wageningen, and they made this into a culture chip. So instead of using agar plates, that you can see on the left, where bacteria are streaked out on a surface and they are growing, you can use this kind of material, which also is subdivided into small culture chips. And you can place this on an agar tray. The nutrients will diffuse through that ceramic or this aluminium oxide material, and the bacteria will grow. But now you can have a lot of different cultures on a agar tray. And if you do this, you can have 1 million small wells on top of this agar plate, and you can actually inoculate a bacteria in every single well if you use automatic inoculation or micro manipulation. So the idea is that you have indicator bacteria that will grow on top of the chip if there is a regrowth potential in the water. So we designed the first version of a flow cell. Oh. And in this flow cell you can see the inlet over here and the outlet over there. There is the water going through it, the flow chamber. Over here is the chip where you can grow your bacteria. This is a seal. There is a material which gives some separation of the different wells before you can, when you want to inoculate the different bacteria. A cover slip, and of course in the end. And actually this was made by Micronit, and we tested this device, the first version. It was reasonable, okay. We had a nice flow of water going underneath the chip, but not everything was as good as it seems. In the first version, we had some tubing leakage, but most of all, it was a little bit too thick to fit on a standard microscope. And what we saw when we grew organisms in this, the visibility of the organisms with a standard microscope was not that good. So we needed to, most likely, we need to stain the microorganisms before we can image the bacteria. Well, this is a bit of a problem, of course, because then you cannot do really online automatic analysis. You still have to add a dye, and that usually means you kill the bacteria. But there are solutions for that. So we came up with a version 2 of the flow cell, which solved most of the problems. There was an improved sealing, so no leakage anymore. We also fitted a temperature sensor in it to correlate the water temperature a little bit with the growth of the bacteria. It is a lot flatter, and you can fix this quite easily on a standard microscope. Also, there was a small minor problem. The cover slip of glass was usually quite difficult to mount, and we had some breakage. But it's also, with this seal, this solved the problem. Well, then it comes actually in using this device. This device is ready this month. So we are going to test this device starting from January in the real water of Water Laboratorium Noord, dus the real purified water. But this technology has already been used by other groups. The groups of Paulien den Hartog and the Tropical Institute in Amsterdam used this chip to measure Mycobacterium tuberculosis. And this organism is a really slow grower and is very difficult to cultivate. By using these chips, they can grow colonies of mycobacteria and already see this within 8 days. And they can actually follow the growth of the bacteria. This is a chip inoculated at day 1, and you can hardly see anything. At day 3, I think at 2, I can't really. Yeah, at day 3, you can see some small dots appearing. And of course, if you do a single measurement, you have no idea if these are bacteria or just particles or some dust which comes on the chips. But because you are following in time, you can actually see your colonies growing. And if you compare this growth rate with the normal growth rate of a mycobacterium, you are quite sure that this is the strain you are looking for, and that the patient is really infected with a mycobacterium. These chips are placed on an agar tray, on a standard growth media for mycobacteria, and every day these chips are placed into an automated microscope. Because this has robotics inside, the chip is always placed at the same position underneath the microscope. So they can actually see that all the dots are really the same dots when they analyze this. And from this, you can make a grow curve. And this is actually exactly what I want to do with the chip on the biofilm. Oh, sorry, on the regrowth potential. Once in a while, you take out the chip, you put it onto the microscope, and you can really follow the colonies which you have inoculated before, and you can follow the growth. And they only grow if there are nutrients available. If you want to determine a specific nutrient, you can also add a lot of other nutrients in excess to the water stream. And only one nutrient is limited. So you can add a lot of carbon, you can add a lot of sulfate and phosphate, and then you only measure the regrowth potential concerning the nitrogen. And if you add nitrogen and carbon and leave out the phosphate, you can determine the phosphate potential of the regrowth. So, of course, you need to detect the bacteria in the beginning. And we did some inoculum tests on these chips and determined the minimum amount of cells which were necessary to be present at T0 before you can detect something. If you do not stain your material, you need a lot of cells inside such a well before you have a nice image. This is done for E. coli, and you need a lot of 2,000 cells. Yeast, the candida, is of course is a little bit bigger, so you need less cells. But overall, you can see if you stain your cells, then the sensitivity goes up. So you need less cells before you can see a nice signal. Well, if you stain with Cyto 9, then you only need 10 to 20 cells at T0 before you can actually see something. Well, I described some of the problems we envision with this fluorescent staining. And also the imaging analysis. These automatic microscope is quite expensive. But there are trends and technologies coming up that you can actually use very cheap microscopes. Yeah, the top part is a €55 microscope. It gives an image on the left. So you need quite a lot of cells. Yeah, you have less sensitivity, but your system becomes way cheaper, of course. If you use a €30,000 microscope, of course your image quality would be much better. To circumvent the problem on the addition of fluorescent dyes, there's also another technology. You can actually use nanobeads to attach to the porous material, and such a nanobead is about an 80 nanometer silica particle with a gold core containing a fluorescent dye. And if you look, it's a bit difficult on this beamer. If you look very carefully, you can see the beads over there. And the beads are attached to the ceramic material, so they don't come into contact with the water. But in case of these beads, if the pH drops because the bacteria are growing, then you will increase your fluorescent signal. So you can still use this technology online in an automatic way without addition of all kinds of different dyes. So the sensor is, the flow cell is being made, I think it's almost done. We will test this material, and I hope to show you the results of this flow cell in a kind of a next conference. I would like to thank Colin Ingham. He's the CTO from Microdish, Gerard Wubbels and Peter van der Maas from the Waterlaboratorium Noord. And of course I want to acknowledge the Samenwerkingsverband Noord-Nederland for funding this project. Thank you very much for your attention.